Hello and welcome to this video on the heritable epigenetic traits that benefit the offspring of otherwise healthy adults. This is an emerging and controversial area of research, as it is very similar to a thoroughly discredited theory of evolution called Lamarckianism. If valid, it would be an important element to note with social movements like fat acceptance, vaping, and even the ironic argument of access to social media platforms like Facebook and YouTube. Mothers who are obese during pregnancy, for example, give birth to children who are more likely to be obese as adults and develop metabolic illnesses such as cardiovascular disease. These changes can carry through a number of generations. The standing theory of inherited genes is that the human genome has a set spread of genes. These can be modified throughout life for that person, but the basic gene is not changed in any drastic manner. This means the offspring of a person will receive a version roughly in line with the copy found in one or both parents. This means parents with a predisposition towards certain diseases or conditions create children with the same disposition. It is a relative constant between generations. The parents can defer this disease as it applies to them for a time through good health and behaviors, exercising, good food, low stress, and similar beneficial things. These would directly modify the genes of a parent in a process called epigenetic modification. This is a series of extraneous factors that do not change the sequence of the gene, but other mechanisms. In genetic terms, it is a short-term and fast modification. This includes the previously mentioned DNA methylation and RNA modifications. These are on top of the DNA. Changes that are not part of the genetic sequence are generally considered more transient and unstable. It is generally thought that as soon as the sperm enters an egg of a mammal, there is a loss of DNA methylation of either parent's chromosomes except where imprinting occurs. What is important to note is that when the DNA crosses over during the genetic shuffle, these alterations have always been thought to be abandoned or lost. This is why the recent and growing body of evidence suggesting that epigenetic changes are also passed on is surprising. To draw a parallel, imagine if a dog breed could be changed through better feeding or care of the animal rather than by breeding of two parents with desirable traits. While not an exact analogy, it is worth reflecting on for the implications of such small shifts and the results. This is distinct from the more readily understood and observed Mendelian genetics. That is, where one pea plant could be bred with another, and you would get a mix of the two. There was no difference if the plant was raised in good soil or bad. It produced a similar hybrid offspring, regardless of its circumstances. You might get diminished yield overall, but you received the same final product. In the context of humans, it rapidly becomes more complicated. There are many more genes, factors, tests, and random variables that show up to mess with your results. Fortunately, we can identify many of these and measure them. In the process of investigating the benefits of exercise, we have a large body of variables that have a known effect. Think of high-fat food and atherosclerosis and heart attack. We also know that even limited amounts of exercise, like half an hour a day, can reduce the risk of liver disease and stroke. Small changes for an individual, which make big changes on the scale of whole populations. This body of evidence is huge and well beyond the scope of this video. What we can describe is what we know of these changes. Let's assume that there are two people, one female and one male. As per past videos, the male has an X and Y chromosome and produces sperm. The female has two X chromosomes and produces the ovum. These are the gametes. Gametes carry a limited copy of the parent's DNA. This DNA is at its base a series of nucleotides. These get translated to make products. The new evidence shows that the gametes do not just inherit this DNA alone. That is where it gets interesting. The gamete only has 23 chromosomes, or half the normal number. The chromosomes are sourced from a larger pool of DNA that is common to the body. The individual cells undergo mutations with age and environmental conditions. These changes are picked up by taking the DNA strand and copying it to split it into another cell during mitosis. This process takes all of the DNA and pulls it apart to each side of the cell, which is then cleaved down the middle. This takes the two halves and just splits them up as is. This produces either two sperm or one ovum. The sperm also carries RNA molecules and regions of the DNA that have been imprinted, which silences that copy of the gene. Some of the ovum genes are also imprinted, although this is more common in the case of a female child who has two copies of the X chromosome. The process of mitosis leads to a gamete with the same changes as the parents have. The two gametes, one from each parent, combine nominally in utero, although it could be IVF or similar. This is shown in a study of prenatal activity. They found an association of DNA methylation and birth weight. They were able to take RNA from the sperm of mice that were physically active, inject this into fertilized egg cells, and identify a pattern within the baby mice that demonstrated higher synaptic activity and advanced learning abilities. This just shows you how these changes, when carried from one generation to the next, affect or at least allow effects to be carried through. It is just one example, and others are available, which you can find in the description. Let's assume that you've successfully passed on your genes. The offspring is now growing in utero. This leads to a second possible source of the epigenetic changes we are discussing. As the fetus grows, it has to undergo an explosion of the cell population in order to develop. As these cells divide, there will be errors in replication. This inserts mistakes, but can also add epigenetic changes. A small modification of how effectively a gene is expressed might be needed to increase the activity, or another gene might need to be downregulated in case it causes malformed limbs. To facilitate this growth and development, the uterus is normally a very hospitable environment for the developing embryo. This is true until you start to add stresses. Things like alcohol, tobacco, and even emotional stress. These can lead to toxic compounds crossing over to the fetus if they meet certain chemical property thresholds. but there'll be more on this in another video. These toxic compounds can cause problems for the developing embryo. One of the best known examples is that of the thalidomide scandal. Thalidomide produced a cell death signal that led to a number of birth defects. In the process of this, the fetus would experience many stresses leading to genetic changes as a result of the damage done. This is not an inherited defect, nor is the next example. Infants of mothers who drink during pregnancy are subject to a slightly different series of events, but with a similar result. That is, they have a more scattered effect that includes abnormal appearance, low weight, small size, poor coordination, low intelligence, behavior problems, and more, that name just a few. These two pathways cause direct chemical damage to the developing embryo, but they are not inherited. These defects occur because of the errors leading to the genes responsible for certain effects being changed. The regulation mechanisms then need to find a new way to control expression, do what they are supposed to do, or otherwise allow the child to develop. This is, in terms of epigenetic changes, the less preponderant avenue. One study tried to understand how a parent could then pass on the genes and have the same effect with epigenetic mutations. Zhu et al. Studied the healthy habits of parental mice and found that they had genetic imprinting even when they used an IVF method. This is the second and more common way, which involves the stress of the environment causing an indirect epigenetic change in the parent, which is immediately carried over to the child. We will keep calling it epigenetic change for simplicity's sake, but be mindful that it is an indirect epigenetic change. As the cells undergo stress, different genes will be activated or suppressed as need be. These will respond to other genes that identify the effect of regulation, allowing them to go up or down. They will then push for the cell to reactivate or decrease the gene's activity, an important step in something as time and process dependent as the cellular proliferation for fetal development. Adding alcohol, tobacco, or even stress from the mother, if she's engaging these habits before pregnancy, will begin to affect the embryo, which snowballs into a bigger issue. This is because the gamete was made with what is effectively damaged material, which means no matter how it is split to form the ovum or sperm, there will be some degree of modification present that is adverse. Having not engaged in any of these three behaviors in particular, they would be less likely but not impossible. According to research from the German Center for Neurodegenerative Diseases in Göttingen and Munich, mice that are exposed to an enriched environment and are able to use their mind and engage in physical activity can give their offspring greater learning ability. This is because they have the same requisite mutations themselves. This clearly highlights two sources of change. One that is from outside sources directly affecting the developing embryo, and one that is inherited. The inherited one will always come from the maternal genetic and paternal genetic line. Each has its own roots, cause, and effect, but ultimately the mechanism by which the offspring receive the epigenetic change is similar, if not fundamentally the same. This is a possible mechanism of gene selection, as each gene would be either reinforced or slowly removed from the population. This is a result of good genes being spread, while negative ones are limited. This positive change is not the only outcome of epigenetic changes. Negative experiences and behaviors also affect offspring. There is evidence of this going all the way back to the US Civil War. Researchers examined the effect being a prisoner of war had both on those who lived that experience and successive generations. They found that those who survived in a Confederate POW camp not only had a far worse life experience themselves, with impaired health, job prospects, and shorter life expectancy, it had an effect on both their immediate children and grandchildren. This was only passed down the male line. These epigenetic changes appeared to be a result of the extremely poor conditions, particularly sanitation and nutrition, that they experienced. They found it led to an 11% higher mortality rate than for a controlled population of non-POW veterans. More rigorous investigation used an experiment with mice. Researchers in 2013 developed the experiment that had intergenerational effects of trauma at its heart. They used an experimental design very much like the learned helplessness experiment. It involved exposing the animal to the scent of cherry blossoms and then shocking it. Every time they exposed it to the scent, the animal would be shocked. Gradually, the animal came to associate the smell with trauma. They then stopped shocking the animal entirely and only exposed it to that scent, where it went through the same process of stress. The animals were then put into a breeding program where they produced several generations of offspring. In order to avoid contaminating the second generation with the first, they were raised by different parents entirely. This ruled out the possibility of the parents teaching the young to be afraid of the smell of cherry blossoms. What they found was that even without that learned behavior, there was some sort of conditioning present through genetic means that led to the animals experiencing stress when exposed to that smell, even though they had not been trained to do so. They did not respond to the same degree, but that does not remove the fact that the second generation was also responding to a stressor they had never encountered. Then there was a third generation bred. These mice also demonstrated a sensitivity to the smell of cherry blossoms. They had never been trained and had effectively no reason to respond, but the design indicates that there was a specific inherited modification that responded to that scent. Not all changes on an epigenetic level are the sole driver of change. Many work in a synchronous way with a deeper level of genetics that constitute the gene's normal function. The two operate together in these complex scenarios to further reinforce an effect. This means that from one generation to the next, beneficial traits may increase or decrease unless reiterated. The converse is true, when traits inherited due to traumatic events may be gradually eroded if resisted, or they may become entrenched through continued exposure. Overall, epigenetic changes are not the strongest of factors predicating one towards a condition or reaction. It is another layer of complexity. What it does show is a mechanism by which inheritance is further complicated, one that can be controlled to a degree, and that will in turn benefit or hinder future generations. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions below.